So, good morning. I could speak in French if you prefer. I mean, up to you. No, nobody raising the hand, so I will go on in English. So thank you for the invitation. I mean, it's the first time I'm participating, and I'm very happy to be there. So I will try to explain what we are doing in the field of epigenetic of grape wine. I think there are not many people in the world working on that. And I think it's very important, especially in this plant, which is clonally propagated, so which means that the genetic identity of population is quite high, and therefore we do have quite a lot of difference in phenotypes and expression. And I think this is quite related, to the topic of terroir that I would like. Okay. So this is the general organization of the talk I will be giving to you. So I'll try first to explain in simple words what we want to speak about when we speak of epigenetics, then relate that to the situation of grape wine, and then I will give a couple of examples of the work we're currently doing to explain why we think that the terroir can be somehow related to the epigenetic memory of plants before having some conclusions. Okay, so first thing, what I was speaking about. So here, this is a quite old work performed on humans, zygotic twins. And so this is a funny image, just maybe I move here so that I can see it sometime. So just to show you that sometimes, real twins, may evolve in phenotypes with time. And this is, something quite puzzling because in real twins, or clones, in other words, the genetic identity remains very high, and except some or a few somatic mutations, change in DNA sequence, you expect them to be very identical. But with time, you can see differences occurring. And this can be reflecting by what we see here on chromosomes. So this is a technique that allows you to look at specific epigenetic marks. I will define them later. And in these young twins here, every time you see it's yellow, it means the two twins are identical on this epigenetic information. But when it's red or when it's green, it means each twin is different. So when you look at 3 years old, you see everything is mostly yellow. So both twins are identical. But when you look at much older twins, which are 50 years old here, then you realize that they are mainly different. So which means that although the genetic information has not been changed, the epigenetic layer of information above it has changed, has evolved. And this can be, in some ways, related to the change in phenotypes. It's not very reactive, I guess. I should move to the next. No, it's not moving. Okay, so let's go to maybe the talk will be a bit longer than expected if we go on this way. So if we go to the exact definition, etymologically, epigenetic means information above the genetic information. So this is a new layer of information. It's not more important, it's not less important, it's an additional layer of information which is initially based on the fact that you may have heritable phenotypes, heritable traits that are not defined by the genetic information, but by another layer of information. And obviously, we do have evidence that in plants, in some cases, variants, like you have here, for example, in tomato, here in snapdragon or on mouse, and now we have evidence in grape wine. We just published a work on that. Some variations, very stable phenotype because these genes have been cloned by classical genetic approaches, which means they are very stable in the population. But when we go and clone the so-called mutation responsible for the phenotype, we find out no difference in genetic information between the red and yellow tomato or between these different flowers or between these two mouths, because the change is not at the genetic level. The change is at the epigenetic information, which differs between both situations, and this change in epigenetic information leads to a very stable change in phenotype. So it behaves like a normal mutations, but it's not a mutation, it's what we call an epimutations. And for example, in grape wine, we have recently shown that the color of the berry, the internal side part of the berry, chair, I don't know not the peel, but inside, which, for example, is found in teinturier phenotypes. I mean, this color occurs because the MIB-A1 gene, which is a transcription factors, exists in different epiallelic state that in some cases allow the expression of the anthocyanin biosynthetic pathway, and in some others does not allow. And so this is not a mutation. It's, again, in grape wine, for example, here an epi mutation is responsible of that. So let's put that in a different way now. So we have the genome. So the genome is what we can call the information of an individual or of a species, which is stored. And if you just look at this, I mean, in the genome, everything is accessible and can be used. But it's not what happened in our cells. What happened in our cells, this information is organized, I mean, very packed, very well, I mean, organized, and it's structure called chromatin, which is an interaction between DNA and histones. And in this case, I mean, the information that was initially accessible might not be all the time accessible because of the packing of the situations. So you can see that as, I mean, a library where you have shelves, initially they should be all accessible, but in a few situations, only a few will be accessible, most will not be. And this is what epigenetic is doing. You will simply limit the access of some genes, and because of that, they cannot be used anymore. This is what is reflected here. And when a gene cannot be used, you can put whatever you want, it cannot be used. In the other situation here, where the gene is accessible, it doesn't mean it will be expressed or it will be used. It means simply it can be used. So if situations are favorable for some different reasons, then the gene might be expressed. So when you look at this, at the whole genome information, this is called the epigenome of a cell, and you have different epigenomes because you have different cells. So which means at the end, when you take the organisms, you have many different specific epigenomes depending of organs, depending of age, depending on developmental stage, and depending also of the environment, which will define the different epigenomes of the plant. Eventually, this may lead to heritable traits, and this is what happens when we have an epiallele, for example. So that's what we are we will try to speak about in the context of grape wine today and see whether we can have evidence that some of the traits that are determined at the epigenetic level can be transmitted and contribute to the so-called terroir. So this, obviously, every table means memory. So what are the memory in a plant? Because we have different types of memory. I mean, we do have different types of memory, short-term, long-term. It is exactly the same for plants. We have different types of memory. There's no cognitive input there. It's only, I mean, biochemistry and cellular and cell biology. So it occurs at the cell level. It's a cell memory. And among the different mechanisms, you have, of course, metabolisms, you have hormones, you have transcription factors, so molecular biology. But probably the most important mechanism for cell memory remains, I mean, the epigenetic information. And then you can look at the plant in different ways. First, a plant is developing, so you have one individual. This individual, I mean, cell will divide, I mean, in a perfect world, so you will have perfect epigenome and things, but this is never what's happening. Usually cells and organisms are put in a complex interaction with the environment, and so this initial perfect epigenome will be modified to adapt. And in some cases, when the plant grows, so which means the meristematic cells, I mean, will divide and so on, some of this information that has been set up by the environment will be maintained. We speak here of somatic memory. It concerns the individual, not the next generation. Eventually, you may have phenotypes occurring, and maybe these phenotypes will be maintained over years. So which means that year after, in perennial plants, like grape wine, some of this epigenetic information will be maintained, and you may have an epigenetic evolution. So this is what we call the transannual memory, which is maintained through meristem during years. But I mean, most also will be lost. I mean, don't you think that it's safe for a plant to forget, because memory is costly. So So this is one aspect. Second aspect is when this plant will reproduce. Obviously, you can have sexual reproduction, and then you have a lot of events occurring, I mean, gamete formation, fecundation, embryo development, and you lose a lot of information, and it's safe also for the plant. But you may maintain some, and that's quite a lot of work on the maintenance of epigenetic information through sexual reproduction. But in the case of grape wine, obviously, I mean, multiplication of the plant, clonal propagation is the main route for creating populations. And here again, we may have a kind of maintenance of the epigenetic information that was provided in the parental plant. So what would be very important in grape wine would be this somatic memory first, this transannual memory because it's a perennial, and also this memory through clonal propagation because it's clonally propagated. And this may define, I mean, the initial start point for a grape wine plant. Okay, so let's put that into the context of grape wine now. So this is a grape wine plant, because I didn't speak of everything, but a plant is not a plant per se. A plant is surrounded by many microorganisms. We speak of the holobiont at the moment, which reconstruct, I mean, the whole environment around the plant, plus the viticulture in the case of grape wine, plus different stresses, global environment. So all this is what will define the plant in a specific state at a specific stage. And all this will determine the change in epigenome that may occur and then the maintenance of this new epigenetic state through plant development. So we are speaking of that. So what do we start? We start from the mother plant. So the mother plant, each of them, from the rootstock and from the scion, they are cultivated in specific areas. They have a specific age, so they have a specific epigenome. And you could even go in details and take the stem and check the different part of the stem and think they may be different also at this stage. So then you graft them. So you have an interaction. I mean, you have a lot of communication through grafting, and you have, I mean, epigenetic information going through both partners coming from the other one that are both influencing each other. So which means just by grafting, in addition to the stress which is caused, you have an information going through which, I mean, redetermine or change in some way the epigenetic information. Then you take those plants and you put them in different fields, and they may forget part of the previous information, maintain part of it, and then they will grow. And after some time, you will arrive to this situation where from the same mother plants have gone through different histories, and they finish up with different epigenomes due to their specific environments. So it's what we have again. I mean, so this situation is not only due to the plant in its environment, but it's also due to the history of the plant, what has occurred before. So how can we address these questions? Because it's very difficult to simply try to see in the very global thing. So we need to dissect a little bit these different processes. And I will not speak of everything here of what we have done. I will mainly focus on this experiment, which is a project we're just starting, which I call Clone Adapt, which is really to try to see from a very clonal population, I mean biological clones, not viticultural clones, which mean coming from one single mother plant. I mean, how do they evolve when they are put in different environments, and what happens when we reproduce them? Then I will speak here of the impact of the microbiome, of the epigenetic information in plants. And then last, I will try to explain one experiment where we provide evidence that we may have also some transannual memory, and all this is contributing to what I've been explaining to you so far. So let's start by this Clone Adapt experiment. The question we are asking is, starting from a very homogeneous material, can we induce epigenetic changes, and are they stable over time? And ultimately, if we can, I mean, can this be related to plant phenotypes? So this is the experimental plan we have been following. So we go to the field, take one plant, I mean, we have selected the plant, propagate them. We don't graft because I don't want to have interaction between both partners. I want to follow what happened at one plant level. And in pots, we put them in two different conditions. Good one, kind of paradise. I mean, everything is provided as they need. And then I mean, very limited conditions for water, heat stress, so combined stress. It's not we don't apply a stress, we put them in conditions where we don't control the stress at all, but we let it, and then we measure what has happened, and we reduce the water supply and the mineral nutrition. And we have been starting this in 2021 for one set of experiments and reproduce it this year. And then we just analyze what happened. And after a while, in some cases, we just take both of those plants and put them again together, just to see how they will evolve. Are they coming close to each other, or are they maintaining them in their differences? And then, but we have just started this, from this situation and this one here, we recreate population, because we want to see whether the difference we have been creating by doing this is maintained in the next generation or not. Okay, so this is just a short summary of some of the data we have been collecting so far. So this reflects, I mean, the stressy situations and the paradise. I mean, I would not like to live in this paradise actually still, but it's good for the plant, not for us. And we just measure a couple of parameters. I mean, I don't show you all of them, but for example, if we look at the main stem diameter, you see that depending on the conditions, I mean, there's a big difference between both plants, which reflect the difference in vigor that we have been generated. And this is the time for the land. So here to average, we have just measured different things, the number of internodes and the average size of internodes. And so we have a big difference in plant size, and this is due both to a difference in internode number and in average size of internode, because they are much shorter in stressy conditions, which is quite classical, but at least we could reproduce that and maintain it. And then, sorry, this slide is a repetition. And then when we take this plant and put them back in the same environment, which is a kind of good one, we see that after 1 year, we do maintain the difference, both considering the main stem diameter and this is, this is a PDF, that's right. So there's another slide normally here, another animation where you see the mean internode size, which is also different. So over 1 year and over 2 years, actually, we do maintain this difference, showing that we have a carryover effect of the previous conditions over 1 or 2 years. So, but this was not a question, we just wanted to see what we have been doing. A question was whether we have created epigenetic differences. So I will not go into details into epigenetic mechanism, I just need one information here. So we have many different types of epigenetic information. One of the most, important, stable, and well-studied is what we call DNA methylation. So on DNA, on cytosines, one specific base of DNA, in some cases you can have a methyl, which is added. And this is an epigenetic information, which is very well studied because it's very stable and carry memory in some cases. So we have techniques to do that, and what techniques use, it's a biochemical approach where we know that when we treat by bisulfite DNA, if a cytosine is not methylated, it will change in another base, a uracil, and this uracil, after some, molecular biology, PCR actually, can be maintained at a timing that we can sequence. But if the cytosine is methylated, it remains a cytosine. It's protected from this chemical activity. So which means at the end, although initially you had only Cs, the Cs that were unmethylated will become T, and those that were methylated will remain Cs. And if you sequence that either locally or at the whole genome level, you can identify precisely which Cs were methylated and which were not methylated. So it's what we do in the lab. And then you can go do that at the whole genome scale and compare its position between situations. In this case, as here, so you can ask a question for one C, was this C methylated or not? And you calculate percentage. It was methylated at 10, 20, 30, 50%. But most interestingly, we can also address that on part of the genome, what we call regions, and you decide the size of the regions. And then when we find differences between both situations, we'll say that this region is differentially methylated. So we call that differentially methylated regions, DMRs, and I will refer to that now for the rest of the talk. So we have been looking at DMRs in different situations. And this is a very brief summary of what we have found here. So let's focus on these plants, stress over 5 years, and here, and look at this situation. So we have had a very stringent screening because we wanted to have very sure regions. And with this very stringent screening, taking, 4 plants in each situation, it's not a lot of plants, but, it's quite expensive, this type of approach, so you cannot do 100 plants in each situation. So we rather analyze different population with a limited number, and then we merge the different data. So we found roughly 400 different regions differentially methylated after 5 years. So which means plants, although the analysis is done at the beginning of the year before any stress application, so when the plant is starting, so this is really the memory of what has happened the year before. So these 400 differences mean that when the plants restart to grow, so before the stress is applied, it remain in the plant, region that remember what has happened the year before. But when, after 3 years, and this is what has happened, we put the plant back together in the same conditions, you see that we recreate a distance depending on the origin. So this plant comes from this, but there are quite a lot of difference with those that has remained in the previous conditions, while those coming from a different origin, so this one here, you see they are very similar. We have only 90, a bit less than 100 differences depending on the origins. So which means that when they remain in different conditions, you maintain in some way, the epigenetic differences, so some memory. But when you put them in the same situations, they converge again quite rapidly. They're not identical. They are different, obviously, but they are less different than those that have been grown for a while in different conditions. So they forget. They maintain part of the memory, but they forget part of the information, which is good news, actually. Okay, so just to conclude about what I've said, so we have been showing that we can generate difference in phenotypes simply by playing on the different growing conditions. And I remind you that the plants are coming from the same mother plant, so there's no difference in the mother plant. The same genetic and epigenetic information initially. And these growing conditions not only create difference in phenotype, but also difference in epigenetic information, memorized, not during the stress, but resuming growth at the beginning of seasons, so before any new stress. And of course, we are now, we have been generating the next generation, and we are studying, the phenotypes as well as the epigenetic information that has been carried over. So this is one first aspect. I'd like to go to now a second aspect that we have been studying, is that the plant is not on its own. The plant is interacting with many different types of microorganisms. Have these microorganisms, so which reflect the final state in the field, have this microorganism an impact on the plant per se, which means at the transcriptomic, so gene expression level, and also epigenetic information level. And this was addressed in the frame of a European project, a PRIMA call, interested in the Mediterranean agriculture. And the idea of this project was to go and to take endophyte from grape wine grown in very arid conditions for decades, with the idea that we have a co-adaptation between the arid condition, the grape wine, and the microbiota around. So, which means the endophyte might reflect the ability of the plant to grow in these very arid conditions. And based on that, we have been isolating this endophyte. You have an example of different endophytes that have been isolating their properties here. I will not detail that, that's not the question, but we do find a few of them, and some are involved in oxygen production, and some have different types of other activities. They can be cultivated. Many could not be. So we had a different approach, which was to graft, the mother plant containing, coming from the arid condition to another plant. But here we'll just present, the cultivable fraction. And then we wanted to transfer this to plants that have been grown in vitro for a while. This was Pinot Noir, not adapted at all to stress, to drought stress, just to see whether we can transfer endophyte into these plants and what would be the consequences, both on the plant and then later to the response to drought stress, which I will not describe today. So here you have different examples of Pinot Noir that has been inoculated with different strains. Maybe you cannot see it properly, but there's, for some of them, a strong effect on the root development. And this can be quantified here, root length. Here you see strain D has a strong impact as compared to the non-inoculated and stronger than the other plants. So we may have, following the inoculation on the endophyte, phenotypical consequences on the plant. So it's not neutral first. And then, so we were quite happy about this result. Then we wanted to investigate a bit more that in detail. So what we did is after inoculation, we took samples after 2 weeks. Obviously, if you take a sample after 2 weeks, you kill it. So which means the 2 weeks and 4 week experiment are independent. We cannot follow up a plant, for such a time. So there are 2 inoculation. One is taken after 2 weeks and one is taken after 4 weeks. So you may have variations due to the fact it's 2 independent experiments. And then we just analyze, the transcriptome, gene expression patterns globally at the plant level and the epigenomes. And so this is what happened at transcriptome level. We use 4 different strains here. And in all cases, when we compare that to a non-inoculated plant, we find differences at the gene expression level. They're not the same. This is after 2 weeks. We can do that after 4 weeks as well, if it wish, blah, blah. Or after 4 weeks. So this is the 2 weeks, this is the 4 weeks. So we do have a specific transcriptomic response for each of the strain. Each of them induce a reaction of the plant, but not the same. And you can see it's not the same when you compare. This is quite a complex graph. Just look at the center. The center reflects, the genes that are the differentially expressed genes that are common to the different strain. They are known. So which means each strain induces a different response. So that's quite important because it's something nobody has described so far. Obviously, you will never find only one strain in a plant. It's also always a consortium, but you need to dissect, to be able to understand. But this was not the point we really wanted to make. We wanted, we also wanted, based on that, to analyze again this DMR between the different plants. And I don't know whether it's like in basketball, when you stop, you remove the time and then you have more time at the end. So, and then we compared here the DMR after 2 weeks. And you see we do have DMRs after 2 weeks in each of the situations. They have very little overlap between the different strains, and they respond in very different ways. So again, we have the same response as those described for the gene expression patterns. Each strain induced a different response. And then when we looked after 4 weeks, each strain has evolved differently into the plant. So which means the impact after 2 weeks, the plant is following is growing up, and then we have a different situation occurring. And maybe after 1 or 2 months, we'll have again a different situations. So it's an evolving situation. And even in some cases, even if we were able to see the strain within the plant at the beginning, after a while, the plant may disappear. Because, the strain, the strain is not, or the plant is not adapted to this very specific strain. So in some cases, in most of them, in 3 of the cases, we do find the strains maintained into the plant, but in one, the strain is disappearing, and still we do have an epigenetic effect, so which is very typical of a memory effect. So all this leads to a complex situation where we can show that strain induce an epigenetic change. This epigenetic change is maintained, whether the strain is or is no more detectable. And interestingly, again, this is very specific. We don't have overlap between the different strains, so each one creates a specific reaction. And of course, now we have the $1 million question. What are the overlap between the change in epigenetic information and the change in gene expression? And of course, as most of my colleagues doing the same thing, we say, oh, we don't have overlap. So which means it's very difficult, and I may explain if you have questions later in more detail, that it's very difficult in most cases to find how epigenetic information influences the phenotype through gene expression. And one obvious reason is simply because we are not looking at the same time scale. Gene expression is a very, photography at a very precise time. Epigenetic is a long-term information, and you may have a gene expression at different times, and if you don't look at the good moment, you don't see it. So that may explain part of the discrepancy we have here. So the main message for this part, if something happened, is that first, and it was not easy to do, first we could transfer on the flight from any type of plant to Pinot Noir cultivated in vitro. Then we have been creating, following this profile, change in gene expression patterns and change in epigenetic information that do not necessarily overlap. And now we are going by inoculating consortia, and this experiment is currently running, trying to see what will be the advantage of a plant that has received this consortia into the response of drought stress. The last point I want to, and I will conclude on that after, is can we show that there is a transannual epigenetic memory? Because this is very important in perennial plants, because this allows you to say that what has happened in year 1 will be maintained in year 2, and eventually impact the behavior of the plant the year after. So to do that, we have developed a totally different experimental system. So this is not what you do in the field, and I wish you don't, because the plants are very much stressed. The idea was to create a nutritional stress to the plant. So we wanted to starve the plant in terms of carbon nutrition, and to do that, what we have been doing is in plants that are at the same age, we had roughly 30 plants of same age, 8 years old, grafted in the same way, and grown in the same conditions. Half of the plants we maintained 12 leaves per bench, and half of the plants, we reduced the number of leaves to 3 per bench. So those were very dramatically starved. We wanted it to be rapid and very efficient. Don't do that. And what happened to those plants here? We realized that, those with 12 leaves, they behave very normally. But those with 3 leaves, we have a very big surprise. Some didn't care at all. We had done nothing. They were very similar to the non-stressed one. But part of them, roughly half of them, were dramatically affected. So first, and we didn't know, we were not expecting that. First, the behavior of plants is very different. The response is very different to the same stress. Some will be highly sensitive to the stress, some will not be. Although they came from the same situation initially, were grown all together during 8 years, and at the end they are different still. So there must be some kind of information carried over that reflect this difference of fragility of the different plants. So, but this was a very nice situation, because here in this experimental system, we could have plants that are controlled all the year long. We could have plants that were stressed 1 year, and that we could happily restress a second year because they were not caring at all. And then this situation is very interesting, because those plants, although they are suffering so much, now we cannot stress them anymore, otherwise we kill them. So we just let them recover. And this situation is perfect, to try to analyze a transannual memory, because you have 1 year of stress, and another thing here allows you to see what has remained at the epigenetic level or of the stress after recovery. And so that's what we did. So we'll just focus on this last situation here. So what we did, we had a stress, and this stress led to this situation, and then we let the plant recover for 1 or 2 years. Each time, and this is very important, we did not sample plants before application of the stress, after application of the stress. We always sampled at the very beginning of the year before application of any stress. So which means the only thing we're looking at is the memory effect. It's not the consequence of the stress, but it's a carry-over effect from year 1 to year 2. And this allows us to say we have a memory or we don't have a memory. So when doing so, what we did is the same. It's quite boring. You don't think I'm always doing the same thing, this epigenetic information, but it's quite informative as well. Okay, so when you look year 1 here, and you analyze, and compared to year 2, the stress has created quite a lot of differences in the epigenetic information. But when you let recover for 1 year, you see these differences compared to the initial situation have decreased. This is what you would expect. I mean, the plant is going back to an initial state. But this was not actually a question, even if it's nice to see that plants are converging again to the initial situation. A question was, is there any of those GMRs created by stress here that have remained here? So we cross these two types of information and could find that roughly 200 DMRs were not creating during the recovery phase, but were remaining from the previous stress that was applied to the plant. So these 200 here reflect the memory effect that was carried over after the stress. After 1 year of recovery, we still do have, part of the information that reflect the initial stress. So this is typically what we're looking for, and now we are digging into this information to find out whether they are related to genes and whether these genes could be involved in the stress response of plants. So I will stop here. So we have shown that we have indeed a memory effect that is carried over for 1 or 2 years, and we don't know how long this may last. I don't think it's safe for a plant to memorize for a too long time. I think because it's costly to maintain or to express genes that are not useful for it. So if the stress is not reapplied, I would expect, my expectation would be that this memory will be erased to come back to a normal situation. And of course, one key point for us is how to relay this epigenetic information, which is memorized to the phenotype of the plant in a global way at the end. So, concluding remarks, so the question was, is that real? Can we relate that to the terroir? I think I have provided evidence that because of the complex interaction created with the microbiome, and this has an impact, with the nutritional state, and this has an impact, and we have some memory effect. I hope I convinced you that indeed, the plants will be different, and this is related to not only the initial history, but also the final state, the final situation where the plant is. And I will skip that because I've been a bit long, and these are the different collaboration of these different projects and the different fundings we may have had. Thanks a lot.